As I have hinted throughout past lessons, we understand that mistakes can happen in DNA replication that can have a profound effect on the processes of transcription, translation, and the resulting functional proteins. So today we are going to look at what happens if we see those mistakes in that process from DNA to RNA to proteins. So throughout this presentation, what I want you to do is get your notes, fill in the appropriate text under the headings, and also when we are finished, there is an attached worksheet in which you will use your notes, use this PowerPoint, and also you may access your online book if you need assistance. And that worksheet is expected to be completed on your arrival to class on Tuesday. So good luck, and email me if you have questions. So as I have hinted through past lessons, as we've been moving through DNA, RNA, and proteins, cells can make mistakes when copying their DNA. These can lead to mutations or proteins that can't actually function or function incorrectly. So mutations are heritable, meaning they can be passed on from parents to offspring. So any changes that actually do occur can actually be passed down from generation to generation. Now we're going to look at two major types of mutations today, the first being gene mutations and then followed by chromosomal mutations. Now gene mutations actually occur at a specific spot in the DNA sequence. Okay, so you can actually point to them, you can say this is where this mutation is occurring. It only affects the production of one protein, which coincides nicely with that definition that we learned of a gene. However, when chromosomal mutations occur, they affect a lot more genes than just one. Okay, so they will occur at the chromosome level, affect multiple genes in different ways, and we will take a look at this as we move through today. So we're going to start by taking a look at our first category of mutations in that realm of gene mutations. So we are going to be on the DNA sequence level. Okay, gene mutations. Now, the first type we're going to look at are known as point mutations. Point mutations simply occur at a single point in the DNA sequence. We can either see substitution, in which a base on the original DNA sequence is actually swapped for another, or we can see insertion or deletion, which is simply the insertion of an extra base in that DNA sequence or the deletion of a base in the DNA sequence. Insertion and deletion are actually known as frame shift mutations. They are essentially a lot more dangerous than substitution. Frame shift mutations actually shift the quote reading frame of that DNA message, and I want to take a look at this so we can put this into context for you. Here on the top we have our original DNA sequence for a normal gene, and what's happening here, as you can see, we're going from DNA to mRNA, so we would be in transcription, the process of transcription. However, we can see right in the middle of our sequence we have 3 adenines in a row. Here in example B, we can see that one of those adenines is being deleted out of that sequence. So what is the effect here? Well, we see on the top that we've got these 3 amino acids that are resulting from the codons in that mRNA strand. Okay, so shift down to the bottom, we can see now that not only the amino acid where the deletion occurred has been affected, every amino acid downstream has been affected. So we will see this on a subsequent slide, but that is the danger of insertions and deletions. They are shifting that reading frame. Every codon downstream of the mutation will be changed, meaning that amino acid has a very high potential to be changed from what it should have been from a DNA sequence that did not have a mutation in it. So this will make a little more sense. We're going to work through it here on the next slide, so let's take a look. So we are going to go ahead and take a little closer look at those 3 types of mutations that we touched on last slide. First we'll look at the substitution. Okay, That occurs when one base of a codon in the mRNA strand is swapped for another. For example, if we begin with cytosine, 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 a substitution could result in something like adenine, cytosine, cytosine. Now, what's important to remember about substitutions is they only affect one amino acid. Okay, because we are not inserting or deleting a base, those codon reading frames will actually stay the same. We will only be affecting this single amino acid in which the substitution occurs. So very important. One base swapped for another, but we are only affecting one amino acid or one codon. So let's take a little closer look at insertion and deletion, those frame shift mutations. In insertion, an extra base is inserted at one point. We can clearly see this in the example here below. If we begin with CTCATA, an insertion could result in something like CTCAGTA. So instead of having these codons marked off here, we will have above stream, yes, we are the same. However, at the point in which the mutation occurs, the reading frame will now be shifted and subsequent codons will all be affected. Very important, shifts the reading frame. So let's take a look here at deletion. Deletion is very similar. Instead of inserting an extra base, we are simply deleting one base from the mRNA strand. So, for example, if we started with CCTAGA, a deletion could result in something like CCAGA. So let's mark off our original codons here. We've got two perfect codons. However, upon a deletion, we are going to affect the codon in which the deletion occurs and everything downstream from that mutation. So very important here, these last two, frame shift mutations, shifting the reading frame downstream from the mutation. Very dangerous, as these can result in proteins that is unable to perform its normal function. So let's take a look at one more example of reading frame mutations or insertions or deletions. This example is going to simply represent the effect of these frame shift mutations. So on the top here we have the original DNA code for an amino acid sequence. This is the resulting amino acid sequence from these reading frames or codons. Okay, a reading frame you can simply think as a codon, those three base sequences. Now, upon an insertion or deletion, these reading frames are changed. The codons are changed. So once again, we began with this amino acid sequence. So the result of everything going correctly throughout transcription and translation would result in this amino acid sequence. However, upon a frame shift mutation, we would end up with an amino acid sequence extremely different, entirely different from what should have occurred from the original DNA code. So you can see the danger of these frame shift mutations of insertion and deletion, resulting in proteins that are unable to perform their original function. Now we also have 4 types of chromosomal mutations that we are going to look at here very quickly. Chromosomal mutations affect the entire chromosome, not just one base on a gene. So remember, gene mutations, very, very specific: substitutions, insertions, and deletions. We are now on to chromosomes that would affect an entire chromosome, which can hold hundreds or thousands of genes. Okay, 4 types we'll look at on the subsequent slides: deletion, duplication, inversion, and translocation. So let's get to it. All right, so let's take a little closer look at these 4 chromosomal mutations that we mentioned on the last slide. The first one we'll look at here is known as deletion. Deletion involves the loss of all or part of a chromosome. So you will be losing multiple genes upon the deletion of all or part of this chromosome. next we'll look at duplication. Duplication produces an extra copy of all or part of a chromosome. Okay, so extra copy, you will extend that chromosome. We'll see that on the next slide. Inversion. Inversion reverses the direction of parts of a chromosome. Okay, so we'll once again see that one on the next slide. And finally, translocation. Part of a chromosome actually breaks off and attaches to another. So one chromosome will be significantly reduced in size and another will be extended or enlarged. So let's move on and take a look. On our final slide of the day, we will be looking at these 4 chromosomal mutations we just looked at. The first one is rather simple, deletion, and it is very easy to see here at the top. So we'll start here with deletion. On the left, we have our normal chromosome, A, B, and C to the left of the centromere, D, E, F to the right. And as a result of the mutation, we see that section B of that chromosome was lost. Okay, once again, a deletion involves the loss of all or part of the chromosome. So relatively easy to see there. Okay, so we'll move on to the second chromosomal mutation, duplication. Duplication produces an extra copy of all or part of a chromosome. So here in this example, we start with our normal chromosome, and after the mutation occurs of duplication, we now have 2 section B's on the left side of our centromere. Okay, so once again, duplication produces an extra copy of all or part of a chromosome. All right, so let me delete this out of here. Okay, and our third type of chromosomal mutation, inversion. Inversion reverses the direction of parts of a chromosome. So we can see the mutation happening here on the left side, and as a result of the mutation on the left side of our centromere, we have AED and CBF to the right of the centromere. So simply we have parts of that chromosome rearrange, reverses the direction of these parts, which can cause issues during gene expression and some other things we will talk about down the line. Okay, so inversion, once again, inversion reverses the direction of parts of a chromosome. And our last chromosomal mutation, our final slide of the day. Here we have 2 chromosomes actually interacting. What I really want you to pay attention to here is the order of the letters. They are sequential. So we begin with A, B, C, D, E, F, G, H, I, J, K, L here on the left side. So they are sequential in alphabetical order. Now, after the mutation on the right, you can see that they have swapped information. This top chromosome, A, B, C, instead of D, E, F, it is now J, K, L, and on the bottom, there is that swapped information on that chromosome. So translocation once again occurs when part of one chromosome breaks off and attaches to another. It is a swapping of information, very dangerous.